The Pharaoh Code, Ancient Egyptian DNA and the Truth Beneath the Wrappings. Geneticists found, when they extracted and sequenced DNA from 90 mummies recovered from the ancient Egyptian site of Abu Sir el-Malek, that the people who built one of the world's greatest civilizations were genetically far closer to the populations of the ancient Near East than to the sub-Saharan African populations that many had assumed them to resemble. And that result, published in the journal Nature Communications in 2017, set off a scientific and cultural debate that is still ongoing. The challenge of extracting ancient DNA from Egyptian mummies had stymied researchers for decades. Egypt's climate, hot, humid in the Nile Delta and subject to seasonal flooding, is among the worst in the world for DNA preservation. Organic molecules degrade rapidly under these conditions, and the embalming substances used by ancient Egyptian mortuary specialists, including natron salt, resins, and oils, further complicate genetic analysis by chemically modifying the DNA that survives. Most attempts before 2010 produced results that researchers suspected were contaminated by the DNA of modern handlers or by environmental bacteria rather than the mummies themselves. The Max Planck Institute for Evolutionary Anthropology in Leipzig developed DNA capture techniques sensitive enough to recover genuine ancient human genetic material from the mummy's teeth and dense petrous bone, the compact bone of the inner ear that has proven most reliable for ancient DNA recovery across many populations and historical periods. The petrous bone's exceptional density makes it the most protective of any bone in the human body for the DNA contained within, and its use transformed the field's ability to recover usable sequences from specimens that had previously yielded only contamination. The 90 mummies analyzed from Abu Sir el-Malek spanned approximately 1,300 years, from the New Kingdom through the Roman period, allowing researchers to track genetic continuity and change across one of history's most consequential stretches of time. Results analysis published by Johannes Krauss, Wolfgang Hark, and their colleagues at the Max Planck Institute established that the ancient Egyptians analyzed in the study clustered genetically with populations from the ancient Levant, the eastern Mediterranean region encompassing present-day Lebanon, Israel, Jordan, and Syria, and with Neolithic Anatolian farming populations from what is now Turkey, rather than with populations from sub-Saharan Africa. The finding did not mean that ancient Egyptians had no African ancestry. It meant that the primary genetic affinities of the Abu Sir el-Malek population across 1,300 years of Egyptian history pointed northeast rather than south. This is consistent with Egypt's geographic position as the northeastern corner of Africa, the ancient crossroads between the African continent and the Eurasian landmass, and the destination of repeated migrations from the Levant and Anatolia during the Neolithic and Bronze Age periods. The second major finding of the study was equally significant. Modern Egyptians carry substantially more sub-Saharan African ancestry than the ancient mummies did. The researchers estimated that modern Egyptians have inherited approximately 8% more sub-Saharan African ancestry than their ancient counterparts, an increase they attributed to intensified trans-Saharan trade and migration in the medieval and early modern periods, including the slave trade, which brought large numbers of enslaved people from sub-Saharan Africa into North Africa over many centuries. The study was careful about its limitations. The Abu Sir el-Malek site represents one geographic location, in Middle Egypt, across a specific time window. It does not characterize the full genetic diversity of ancient Egypt, whose population ranged from the Mediterranean coast to Nubia, and whose history included centuries of Nubian rule. DNA analysis of royal Egyptian mummies, published separately from the Abu Sir el-Malek study, produced findings about the genetic lineage of specific pharaohs that generated both scientific interest and considerable controversy, particularly the results associated with Tutankhamun and his family. In 2010, a team of Egyptian and international researchers, led by Zahi Hawass, published the results of DNA and CT scan analysis of 11 royal mummies from the New Kingdom period, including Tutankhamun, his probable father Akhenaten, and several individuals whose identities had been uncertain for decades. The Y chromosome analysis of Tutankhamun and the male mummies identified as his probable paternal relatives showed haplogroup R1b, a lineage most commonly associated today with Western European populations, present in approximately 70% of men in Britain, Ireland, and France. The R1b finding generated headlines claiming European or Caucasian ancestry for the pharaohs, but geneticists were quick to contextualize the result. R1b is not exclusively European. It is present at significant frequencies in the Caucasus, Anatolia, and parts of the ancient Near East. At the time of the 18th dynasty, the geographic distribution of R1b was different from today, and its presence in Royal Egyptian DNA is most parsimoniously explained by Near Eastern ancestry, not any connection to modern Western Europe. The finding is a reminder that haplogroup labels, which describe lineages defined by modern population distributions, can be misleading when applied to ancient individuals who lived before those modern distributions were established. What the Tutankhamun DNA study confirmed with more certainty was a pattern of close kin marriage within the royal family. The genetic analysis identified evidence of inbreeding across multiple generations, consistent with the historical record of sibling and half-sibling marriages within the Egyptian royal house, practiced to maintain the divine bloodline and consolidate inheritance. Forensic DNA analysis published as part of the 2010 Royal Mummy Study resolved several long-standing questions about the maternal lineage of Tutankhamun, and in doing so, identified the remains of individuals who had previously been unidentified, including the probable mother of Egypt's most famous boy king. A mummy found in tomb KV35 and known before DNA analysis simply as the younger lady was confirmed through genetic comparison to be both a daughter of Amenhotep III and Queen T, and the mother of Tutankhamun. This made her a sister or half-sister of Akhenaten, who was identified as Tutankhamun's father, meaning that Tutankhamun was the product of a sibling marriage, a practice documented in the historical record of the 18th dynasty, and consistent with the genetic evidence of inbreeding visible in his skeletal remains. Queen T, Tutankhamun's maternal grandmother, was identified from a mummy found in the same KV35 cache. Her DNA matched genetic profiles consistent with Egyptian rather than foreign ancestry, contradicting older theories that she had Nubian or Mitanni origins. The mummy of Nefertiti, Akhenaten's chief wife and Tutankhamun's stepmother, has not been definitively identified, and no genetic analysis has confirmed which unidentified female mummy in the Valley of the Kings corresponds to her. What DNA could establish, if her mummy is ever identified and analyzed, is her geographic ancestry, specifically whether she was of Egyptian, Mitanni, or other Near Eastern origin, a question historians have debated for over a century without resolution. CT scanning of Tutankhamun's mummy alongside the DNA analysis produced a revised physical portrait. A young man with a slightly cleft palate, a club foot requiring walking canes, and evidence of multiple malaria infections, all consistent with the health consequences of close kin marriage and with the collection of over 100 walking sticks found in his tomb. Archaeological and forensic evidence found in Egyptian burial contexts, examined through the combined lens of DNA analysis, CT scanning, and traditional Egyptological scholarship, documents a mortuary practice of extraordinary complexity, one whose biological record preserves not just physical remains, but traces of the beliefs, fears, and social hierarchies of a civilization that spanned 3,000 years. The mummy, designated Unknown Man E, found in a cache of royal mummies at Deir el-Bahari and known informally as the screaming mummy because of the contorted expression preserved by the embalming process, was identified through genetic analysis as almost certainly Prince Pentawera, a son of Ramesses III implicated in the harem conspiracy, a plot to murder the king and place Pentawera on the throne that is documented in the Judicial Papyrus of Turin, one of the most important legal documents to survive from ancient Egypt. The papyrus records the trials and convictions of the conspirators, including the queen who organized the plot and Pentawera himself, who was condemned to take his own life rather than face a formal execution, a distinction that carried religious significance in Egyptian belief about the afterlife. The mummy's unusual treatment supports the identification. Rather than being embalmed in the standard manner, with the body relaxed and the face composed, unknown man E was wrapped in a goat skin, an impure material in ancient Egyptian religious tradition, with his face left in the anguished expression. The use of ritually polluting material in the burial was a deliberate post-mortem punishment, denying the deceased the standard passage into the afterlife. The DNA analysis, which showed a familial relationship consistent with a son of Ramesses III, gave this interpretation a biological foundation. The practice of retaining the heart within the body while removing other organs for canopic jar storage reflects Egyptian belief that the heart was the seat of intelligence and soul, the organ that would be weighed against the feather of Ma'at in the judgment of the dead. Skeletal and genetic evidence found at the workers' settlement adjacent to the Giza Plateau, excavated systematically since the 1990s by a team led by Egyptian archaeologist Zahi Hawass and American archaeologist Mark Lehner, has produced a biological profile of the people who built the Great Pyramid and the monuments surrounding it that contradicts the most persistent popular misconception about ancient Egyptian construction. The workers buried in the cemetery adjacent to the pyramid complex were not enslaved foreigners, as a reading of the Hebrew Bible's Exodus narrative might suggest. DNA analysis of the skeletal remains shows they were Egyptians, with genetic affinities consistent with the broader ancient Egyptian population identified in the Abu Sir el-Malek study. Isotope analysis of their bones shows signatures consistent with childhood spent in the Nile Valley, not in foreign territories. The physical evidence from the skeletal remains tells a story of demanding but not exploited labor. Healed fractures show evidence of sophisticated medical treatment. Broken bones were set and splintered, and the individuals survived to continue working. Skeletal markers of heavy physical activity are present throughout, but so are indicators of adequate nutrition. The animal bone deposits found in the workers' settlement, including the remains of cattle, sheep, and goat, in quantities suggesting regular meat consumption, indicate that the pyramid workers were fed substantially better than most ancient Egyptian commoners. The scale of the logistical operation required to feed or house and provide medical care for the estimated 20 to 30,000 workers who lived at Giza during peak construction phases implies a level of administrative and crocodile mummies from the temple site of Kom Ombo have also been analyzed. DNA results revealed two genetically distinct ancient crocodile species, Crocodylus niloticus, the Nile crocodile still present in Africa, and Crocodylus succus, a species previously known only from ancient texts. C. succus appears to have been specifically associated with temple worship in Upper Egypt, and the discovery of this distinct species from mummy DNA alone is among the more unexpected results of ancient animal genomics. Population genetic research on ancient Egyptian samples from different periods and locations documents successive waves of migration and genetic influence that transformed the Egyptian population across 3 millennia. A layered history of conquest, trade, and cultural exchange that the DNA record preserves in a form that no historical text fully captures. The Hyksos, the foreign rulers who controlled Lower Egypt during the Second Intermediate Period, roughly 1650 to 1550 BCE, have long been debated regarding their origins. Egyptian texts describe them as invaders from an unspecified foreign land. The name Hyksos derives from an Egyptian phrase meaning rulers of foreign lands. Ancient DNA analysis of individuals from Tel El Dabba, the Hyksos capital in the Nile Delta, published by a team from the Austrian Archaeological Institute and the Max Planck Institute, found that the Hyksos rulers carried genetic profiles consistent with Levantine and broader Near Eastern populations, confirming a foreign geographic origin. The study also found evidence of earlier migration waves into the Delta region before the Hyksos rose to political power, suggesting that the population of the Delta had been receiving Near Eastern migrants for generations before the political transition that Egyptian records frame as a sudden conquest. The Hyksos introduced the horse-drawn chariot, composite bow, and bronze weapons to Egypt, military technologies that the Egyptians subsequently adopted and used to expel the Hyksos and establish the new kingdom. The Ptolemaic period, the 3th-century dynasty founded by one of Alexander's generals and ending with Cleopatra VII, provides a distinct genetic layer. Cleopatra's ancestry was Macedonian Greek on her paternal side through the Ptolemaic line, and the dynasty practiced sibling marriage following Egyptian royal tradition. Whether she had any Egyptian ancestry through her maternal line is not established, as no confirmed Ptolemaic royal mummy has been successfully analyzed for DNA. Forensic genetic research applied to mummies held in museum collections worldwide, many of them acquired during the 19th and early 20th centuries, in circumstances that would not meet modern ethical or legal standards, has produced identifications and attributions that are changing both our understanding of specific individuals and the legal framework governing how ancient human remains are held and studied. The Egyptian Museum in Cairo holds the world's largest collection of royal mummies, and systematic DNA analysis of the collection has been ongoing since the early 2000s under the supervision of Egyptian authorities. The identification of specific individuals from the KV35 and Deir el-Bahari caches has established family relationships that the historical record had left ambiguous, including the identification of the younger lady as Tutankhamun's mother and the genetic confirmation of Queen T's remains. The question of the pharaoh's curse, the supposed supernatural deaths of those who disturbed Tutankhamun's tomb in 1922, has been examined through the lens of ancient microbiology. DNA analysis of organic material from Egyptian tomb environments has identified ancient strains of Aspergillus flavus and other fungi that can cause serious respiratory illness in immunocompromised individuals. The causal connection between tomb exposure and specific deaths among the Carnarvon expedition members is not established by the evidence, but the biological mechanism for illness is real. The ability to reconstruct facial appearance from ancient Egyptian DNA, combining genetic variants associated with skull morphology, skin tone, eye color, and hair type, has produced probabilistic reconstructions of several pharaohs that are consistent with, and in some cases more detailed than, their mummy portraits and sculptural representations. Scientific synthesis found across the ancient Egyptian DNA literature of the past decade presents a picture of one of history's most consequential civilizations, not as a static, racially defined entity, but as a dynamic and genetically diverse population whose composition changed substantially across 3,000 years of history, and whose legacy is carried in the genomes of millions of people across North Africa, the Middle East, and the Mediterranean world. The core conclusion of the 2017 Max Planck study, that ancient Egyptians were genetically more similar to ancient Near Eastern and Anatolian populations than to sub-Saharan African populations, does not diminish Egypt's African identity. Egypt is geographically, culturally, and historically an African civilization. What it means is that the genetic history of the Nile Valley is complex, layered, and connected to both Africa and Eurasia in ways that no simple racial categorization captures. The Ancient Egyptian Genome Project has contributed to a broader revision in how scientists and the public understand the relationship between genetics and identity. Ancient DNA repeatedly demonstrates that the populations of the past were in motion, migrating, mixing, replacing, and being replaced, in ways that make the concept of a genetically pure ancient people untenable in almost every historical context examined. Egypt is no exception, and its story is more interesting for it. What the mummies preserve is not a racial category, but a civilization, a specific, extraordinarily accomplished human project that drew on the talents and labor of people from across the ancient world, left a physical legacy that has survived 4,000 years and is now yielding its biological secrets, one genome at a time.